Hello, AP Bio. This is part 2, video 2 from chapter 3. If you recall from video 1, chapter 3 is on carbon and the metabolic diversity of life. In the second video, we're going to go through the 4 types of macromolecule. I should go ahead and warn you now, this half of the chapter has a lot of just detailed information. There's lots of just dense stuff to learn. You're probably going to want to pause this throughout and copy down things that we're going through pretty quickly because the details here are all very important. Okay, so we're going to go through these one at a time. So the first class of macromolecule is called carbohydrates. Carbohydrates are essentially sugars and their polymers. For each of the 4 types of macromolecule, we're going to discuss what the monomer is, what the building block is, and then what the polymer is. So for carbs, the monomers are called monosaccharides. Mono means one, saccharin refers to sugar. The sort of most basic is glucose, and we've seen glucose before. Glucose is C6H12O6. If you divided each of the subscripts by 6, you would basically get CH2O, which is where the word carbohydrate, as in carbon and water, hydrate comes from. But carbs they're not just carbon mixed with water. They have pretty elaborate structures sometimes. So the 3 monosaccharides you should know is glucose, galactose, and fructose. All right. You think of carbs as being sweet. Not all carbs are sweet. Most of the sugars that are in things that we eat is based upon high fructose corn syrup, which fructose is pretty sweet. Glucose is not that sweet. It's more like if you had like a very kind of like a cracker that was only like mildly sweet. That's more what glucose tastes like. If you look at the structures of glucose, galactose, and fructose, they're either 6-membered rings or 5-membered rings. Okay, cool. So if I remember how I mentioned this in video 1, that sugars like glucose, when they're aqueous, when they're dissolved in water, they linearize. We've already discussed this. Here's the linear form of glucose, and here's the alpha in this case, that's the alpha form, right? Form of glucose. Okay, cool. So the linear forms of glucose, galactose, and fructose, you do not need to memorize these. You don't really even need to know it. Just understand that the ring structures of carbs can linearize when they're in water. Okay, so if I take 2 monosaccharides and bind them together, you get what's called a disaccharide. The bond between disaccharides, I'll put the slide back. It's called a glycoside bond or a glycosidic linkage, like what's shown here. If you take 2 glucose molecules and combine them, that's called maltose. A glucose and a galactose is called lactose, which is milk sugar. And a glucose and a fructose is sucrose. Sucrose is also decently sweet, kind of like fructose. Okay. And again, this is the same kind of dehydration or condensation reaction that we saw in video 1, where you form a new bond, in this case a glycosidic bond, and give off water. This is called a 1,2 glycosidic bond. Each carbon is numbered with a number. So this is carbon 1 of the first monosaccharide. This is carbon 2. So because the bond is between these 2 carbons, you call it a 1,2, in this case glycosidic bond, because it's a carbohydrate. Polysaccharides, a few hundred to a few thousand monosaccharides linked together. These are going to be things like starch, glycogen, cellulose. In terms of storage, usually it's going to be providing energy, like starch or glycogen, or for structural support, like cellulose in plants. Okay, so let's go through these quickly. So a starch, it's a storage polysaccharide. It's basically just hundreds, if not thousands, of repeating units of glucose. The simplest form is called amylose starch. When you eat a piece of bread, that's anything that's very, very starchy, cereal, bread, any kind of grain. There's an enzyme in your saliva called salivary amylase. I combined 2 words for a minute. Salivary amylase is an enzyme that breaks down amylose, which is starch. So that piece of bread or that cracker, just let it sit in your mouth. Your saliva will start to dissolve it. Glycogen is a form of starch, kind of nicknamed animal starch sometimes. Number 1, starch, that's formed by plants. Number 2, glycogen is formed by humans and some other types of vertebrate animals. Glycogen, so when you eat sugary meals, that obviously increases your blood sugar, but your body will store that, eventually it might store it as fat, but it's going to have a short-term supply of what's called glycogen, which is mainly stored in your liver, in your muscle cells. So if your body needs a quick fix of sugar and your blood sugar is low, your body will start breaking down your glycogen stores in your muscles, which makes sense because your muscles need lots of energy, and in your liver. If you look at this diagram, so here you see a bunch of glucoses bound together. This is the structure in starch. This is the structure in glycogen. They look very similar. Glycogen is more branched. See how it's branching off? Starch doesn't branch as much. Cellulose, which we'll get to on the next slide. Cellulose, remember we were discussing the alpha and the beta conformation of glucose? So do you see how in starch and in glycogen, the oxygens always point down, right? And the oxygen in the 6-membered ring is always the top right. So cellulose, if you notice, the oxygens alternate. That one points down, that one points up, that one's down, that one's up, that one's down, that one's up. And the oxygens in the rings are either in the top right corner or the bottom right corner. So basically the difference is, the enzyme that makes starch or glycogen or cellulose, whether or not the alpha version of glucose or the beta version of glucose is what goes into the enzyme, dictates whether it's starch or glycogen, plant versus animal, or cellulose. And long story short, I'm getting ahead of myself, but you don't have enzymes that can break down cellulose. You can't break down this conformation of the polysaccharide. You can break down starch. You can break down glycogen. You can't break down cellulose. It's fiber. You don't break it down, it just goes right through you. And cellulose is what plants use to make their cell wall. It's a tough, rigid outer covering, makes plants grow upright. It's still a glycosidic linkage, but it's one's between the alpha carbons and, the alpha structure of glucose, the other one's between the beta structure of glucose, which, just to remind you here, remember they can go back and forth via the linear form. Once the polysaccharides form, you can't go back and forth once you've made the polysaccharide. It's kind of locked in place. This one shows the alpha conformation. This would be in starch or glycogen. This shows the beta conformation. This picture's from a different textbook, so it looks a little bit different in format from the ones in your textbook. But long story short, you have enzymes that can break down the alpha version. So bread, glycogen, you're good. You don't have the enzymes that can break down the beta conformation. So when you eat lettuce, when you eat things that have lots of cellulose, a piece of celery, you don't digest that. It's called roughage. It's good for you because it keeps you regular. It keeps you, going potty on a regular basis, that's good for you. If you get, stopped up, that's bad for your body. So it's good for you nutritionally because it keeps you regular, but it doesn't, you don't digest it. It's just fiber. Eating a good salad gives you lots of fiber, makes you go. This slide just says all the things that I just said. Some animals, like cows, that can digest, cows eat grass, they can digest cellulose, either have adaptations in their digestive system that allows them to do that you don't have, or it might have bacteria or some kind of microbe in their guts that helps break down the beta conformation of cellulose. But you cannot. It goes through you as insoluble fiber, keeps you regular. One note, another polysaccharide of glucose is called chitin. This word is not chitin. People like to mispronounce it. It's chi, like the Greek letter chi. Chitin is a polysaccharide that fungi use in their cell walls, and arthropods use it in their exoskeletons. So if you stomp on a roach and it cracks, makes a crunchy sound, that's due to chitin, which is also a carbohydrate. Okay, so that's all about carbs. All right, we're 1 down, 3 to go. Lipids. Lipids, by definition, are things that do not mix with water. So fats, oils, wax, things like that. Their primary function in biology is to store excess energy. You can eat all the carbs you want. If you eat too many carbs, your body will convert the carbs as fat because fat's a more efficient way of storing excess energy than is carbs. Things that they advertise as being fat-free, that's great, but if they're loaded up with lots of carbs, your body just turns that into fat. So it might be fat-free in what you're eating, but your body makes it your body turns it into fat. Examples: fats, phospholipids, and steroids. We'll get to all those in just a minute. So, okay, this gets complicated. So the general form of a fat is called a triglyceride. Let me look at something real quick. This is just that same picture bigger. Okay, so glycerol is this molecule here that's highlighted in this kind of tan color is called glycerol. It's a very small molecule. It's a 3-carbon molecule. Each carbon has a hydroxide group, an OH group, and the rest are just hydrogens. Each OH group can be bound to what's called a fatty acid. If you look at this fatty acid, this kind of pumpkin color, so they're long chains, long hydrocarbon chains that could be hundreds, if not thousands, of carbons long. The end is a carboxylic acid group, which is why it's called a fatty acid. This is obviously going to be very, very nonpolar because CH bonds are nonpolar. They don't mix with water that well. So a long chain of just CH bonds is going to be pretty nonpolar. So basically what you do is you do 3 dehydration reactions. So here, to make one fatty acid, to make one triglyceride, you have one glycerol. This is a glycerol. A fatty acid there, which is shown, a fatty acid here, which is not shown, and a fatty acid here, which is not shown. So to make one of these big triglycerides, you're going to produce 3 water molecules. I just used the word. Triacylglycerol or just triglyceride is what you call this structure right here. The bond is called an ester linkage. It's not that important that you know that, but remember the sugars were formed by glycosidic bonds. Fats are formed by ester linkages. Okay, so do you see in this picture right here, the bonds between all of the carbons are single bonds? What would happen if this bond right here, between these 2 carbons that I'm pointing to, what if I made that into a double bond? If I did that, the carbon would have too many bonds, right? So if I made this into a double bond, you'd have to lose 2 of the hydrogens, okay? And if you look, I'll put the slide back. If you look at like this diagram here, I have a double bond, which means, you know, this carbon only has room for 1 more hydrogen, as does that one. So saturated versus non or unsaturated fats. So saturated means it has all the hydrogens that it can possibly have. It has no double bonds. So all of the fatty acids in this picture are saturated. There's no double bonds. There's lots of hydrogen, right? If I were to add a double bond, you're going to lose some hydrogen, like in this one right here. That's called an unsaturated fatty acid because it doesn't have the maximum number of hydrogens because it has double bonds, 1 or more double bonds. Things that are saturated fatty acids are usually solid at room temperature, like a fat. Things that are unsaturated are usually liquid at room temperature, like an oil. And the reason is when you add double bonds, it kind of causes the fatty acid chain to become kinked. See how it's kind of bent? And when you have kinks, the more kinks you have in the fatty acid chains, it sort of spreads things apart. You can't pack them as tight. So this is showing olive oil versus butter. When they're all saturated, you can pack them really tight, which makes it more dense, which makes it a solid. When you add kinks, it makes it spread out more. You can't pack it as tight, so it's more fluid. Saturated fatty acids are worse for you than our unsaturated fatty acids, which are better for you. Obviously, cooking with oil is better than cooking with butter or margarine. Okay, this is a super important concept. This is something that you for sure need to know backwards and forwards. Phospholipids, another example. If I took, let me go back. If I took triglyceride and got rid of the top fatty acid, just get rid of it and put a phosphate group instead, you get what's called a phospholipid. Phospholipids are interesting because they're kind of schizophrenic molecules in a way, because, you know, lipids are hydrophobic. They don't like water, but a phosphate group has those negative charges, remember? Here you can see negative charge here. Phosphate groups do mix with water. So this molecule, the word is amphiphilic. Half of the molecule or part of the molecule likes water and part of it doesn't like water. The head, the phosphate head, is hydrophilic. The fatty acid tails are hydrophobic. So if I mix these things with water, like, what's going to happen? Well, what happens is they sort of self-assemble into a bilayer. This obviously will bring us to the cell membrane in a later chapter, where I have 2 layers of phospholipids, and the phosphate heads, which like water, face out toward the water, and the fatty acid tails, which don't like water, they repel water, they self-assemble together in a bilayer, okay? This slide's what I just said. The phosphate heads like water, hydrophilic. Fatty acid tails don't like water, hydrophobic. Steroids are another type of lipid. We don't need to go into that much detail. This was, we also saw estrogen and testosterone in a previous video, how they were four-membered rings that were fused, or four fused rings in one structure. I said that incorrectly. It's four rings that are fused. Cholesterol is another type of lipid, which we're going to revisit cholesterol when we do the cell membrane. High levels of cholesterol can lead to cardiovascular disease, which again we'll come back to. Okay, 2 down, 2 more to go. So proteins. Proteins, wow. Carbs we said were sugars in their polymer storage and structure in some cases, easy definition. Lipids repel water, easy definition. Proteins don't have an easy definition. Proteins do, they're like the workhorses of macromolecules. They do defense, storage, transport, communicate, movement, structural support, you name it, proteins do it. Just to go through some examples, so proteins are enzymes. Enzymes speed up chemical reactions. In chapter 6, we'll come back to enzymes. Enzymes are proteins. Your immune system, antibodies. Antibodies target bacteria, viruses, things that your body should break down. Antibodies are proteins. Storage proteins, like the white of an egg, those are proteins. Transport proteins, this is an example of a protein embedded inside a cell membrane that allows things to go through the cell membrane. Transport proteins, you guessed it, are proteins. Hormones can be proteins. Receptor proteins, this is a nerve cell. This is showing the little red things are called neurotransmitters. They're sending nerve cells between cells. The receptors on the second cell are proteins. Your muscles, the actin and myosin, contractile proteins of your muscles, those are proteins. Keratin, your fingernails, your hair, structural proteins in your body for support, those are proteins. So proteins do a lot, all right? What's more important, I think, to know about proteins is how they're made. So proteins, the monomer of a protein is called an amino acid. Yeah, I think you probably did this in 9th grade. Amino acids, remember the monomer of a carb was a monosaccharide. The monomer of a protein is called an amino acid, and there are 20 amino acids that that make up the alphabet of proteins. It's kind of a cool number because our alphabet has 26 letters. The alphabet of proteins has 20 letters. 10 of them your body can make, 10 of them you have to get from your diet. If you look at an amino acid, and this is super important, this is a basic amino acid. It's called an amino acid because, okay, so there's a central carbon, right? There's the amino group, which is an NH2 group. And then there's a carboxylic acid group, which is the C double bond O single bond OH group. So hence the name amino acid. The R group, the side chain for the 20 amino acids, this part's the same, that's the same, that's the same. The R group is what's different for the 20 amino acids, and we're going to go through them in a minute. Actually, we'll go through them right now. Now, these next couple slides, you do not need to memorize these. You don't need to memorize the names. We're going to go through them just so you're familiar with them, but don't let this overwhelm you. So this slide is showing 9 amino acids. Notice they have names: glycine, alanine, valine, leucine, isoleucine, methionine, phenylalanine, tryptophan, proline. Again, you don't need to know the names. Each one has a 3-letter abbreviation and a 1-letter abbreviation. As long as you know that VAL is an amino acid and understand what that is, you're fine. So this slide shows ones that are hydrophobic, and they're hydrophobic because of the R groups. The amino side, the carboxylic acid side, honestly not that interesting right now. It's the R group that is interesting. If you look at the atoms and the R groups, I see lots of carbons and lots of hydrogens, which remember CH bonds are nonpolar. Okay, so those are the hydrophobic ones. So let me ask you a quick, actually, no, let me come back to that question. So these are the side chains that are hydrophilic. Notice I see lots of OH groups: serine, threonine, cysteine, tyrosine. asparagine, glutamine. Actually, lots of things that are polar, right? Some sulfur, some oxygen. These 5 are still polar. They're still hydrophilic. They're superpolar because they're charged. Aspartic acid, glutamic acid, lysine, arginine, histidine. These 2 have negative charges and they're acidic. These 3 have positive charges and they're basic. So based upon what amino acids are in your protein, it could be polar, nonpolar, acidic, basic, charged, positive, negative, not charged. The R groups are what give the protein its flavor. They're going to be what gives the protein its structure too. So quick question. So let's say I had a protein. Let's go back here. That was 1,000 amino acids long. And let's say that I made a mistake. And where there was supposed to be a glycine, I accidentally had a mutation, got a sunburn, had a mutation in my DNA. And instead of glycine, it got replaced with alanine. All right. Is that a big difference to replace that one with that one? Well, it might be. Okay. It's the wrong amino acid. So it could be a big deal. But more than likely, replacing that with that is not that big a deal. Okay. One small nonpolar amino acid replaced with another small nonpolar amino acid, likely not a big deal. Now, let's say that the amino acid is supposed to be glutamic acid, this bad boy right here, and I made a mistake and I accidentally replaced glutamic acid with valine. So I replaced a large acidic, negatively charged amino acid with a decently small, neutral hydrophobic amino acid. That's going to be bad, right? Why that's bad, we'll see in a minute. But that R group is totally wrong. The protein's going to fold incorrectly. The example I just gave you is what causes sickle cell anemia. And the protein hemoglobin, which is a protein that carries oxygen through your body, which is super important. It's a huge 4 subunit globular protein. But there's one mistake where there's supposed to be a glutamic acid, got replaced due to a mutation with a valine. And those are way different amino acids, so that's going to be bad. Okay? We'll come back to sickle cell anemia in a minute. The bonds that form proteins are called peptide bonds. Anything pep or peptido in biology refers to protein. Notice, look at this. It's a dehydration or condensation reaction. You give off water. Note that proteins have what's called a C-terminus and an N-terminus. This side of this could be 1,000 amino acids, but one side is going to end in the amino group of the first amino acid, which is the N-terminus. The other end is going to end with the carboxylic acid group of the last amino acid. That's the C-terminus. Okay, so a protein's structure and function. The structure tells you its function. Proteins are big molecules that fold. If you look at these 3D models from your book, like proteins aren't just long strings. The amino acids are in a sequence, but the protein is going to coil and pleat and fold back on itself, and the protein's shape is what determines its function. Sickle cell anemia is bad because the hemoglobin has the wrong shape. It has that sickle-celled shape, which means it doesn't carry oxygen as much. The cells get clogged easily at joints. They get blocked. It can cause pain, cause fatigue, because the protein is the wrong shape. So what we need to go through now, this is just a picture. This is an antibody. This is a protein from the flu virus. See how they bond here together? If the antibody was the wrong shape, it wouldn't bind to the flu virus and it wouldn't destroy it. So there's 4 levels of protein conformation. Primary, secondary, tertiary, quaternary. The little 1 with the little degree sign is primary. That's what that means. Usually you learn that in math class. So the primary structure is just the order of the amino acids. Glycine, proline, threonine, glycine, threonine, glycine, glutamine, serine, lysine. It's just the order. The order for the protein is coded for in your DNA, which obviously we'll discuss, transcription and translation in a later chapter. That this is not for today. Now, proteins aren't shoestrings. They're not long, just linear molecules. They're going to fold. But the primary sequence obviously determines how it folds. The secondary structure, there's 2 examples. There's what's called an alpha helix, alpha helices, which are like the old-timey phone cord. It's like a single helix, like a spiral ribbon. And there's what are called beta pleats, which is kind of like this number, like corrugated cardboard. These 2 structures are formed by interactions between the amino group and the carboxylic acid group. The secondary structure does not involve the R groups. It involves just the backbone. And a protein could have, multiple alpha helices, beta pleats in different parts of the molecule. If I go back a second, like in this picture, I see an alpha helix there, an alpha helix there, an alpha helix there. I don't really see beta pleats in this picture. But a protein can have multiple of both. Of note also is they're formed by hydrogen bonds between amino groups and carboxylic acid groups at regular intervals. So, hydrogen bonds they're weak bonds, they're not strong, but if you have a lot of them, they can have a big impact. If I break the hydrogen bonds, you're going to mess with the protein's shape. So breaking hydrogen bonds is usually a bad idea. The tertiary structure, so the primary was the sequence, the secondary were alpha helices or beta pleats. The tertiary is when a protein folds back on itself. So you could have amino acids that are hundreds of units apart, but if this one's negatively charged and this one's positively charged, it could fold back on itself and clump. These are caused by interactions between R groups, hydrophobic. So if I have a protein that's lots of hydrophilic amino acids, and amino acid 1 and amino acid 50 are both hydrophobic, they're going to come together. They're going to clump together. Ionic would be positive and negative. Hydrogen bonding, we've discussed. Disulfide bridges. Cysteine, the amino acid cysteine, if you'll humor me, cysteine has sulfur in it, remember? Sulfur likes to bind with itself to form what are called disulfide bridges, which are basically bonds. This is a disulfide bridge right here between two cysteines, and they can be way far apart. But because they attract, the protein folds back. So here I see a hydrogen bond, hydrophobic interactions, an ionic bond, a disulfide bridge. All these in conjunction cause the protein to fold. The quaternary structure, the 4 with the degree symbol, this is just if a protein has more than one subunit. So say I have 2 proteins, they're different polypeptides, like they're made separately, but they come together and bond. And that protein has 2 subunits. That's the quaternary structure. Hemoglobin, for example, has 4 different subunits to form one hemoglobin molecule. And those could be ionic, polar, nonpolar, you name it, any kind of reaction or attraction that brings the subunits together. So primary was a sequence, secondary were alpha helices, beta pleats, tertiary, it folds back on itself, quaternary, different subunits. This is showing collagen, which is in your hair, your skin, and you can see it's in this picture, you can see 3 different subunits. Hemoglobin, there's 2 alpha subunits, 2 beta subunits. This just reviews primary, secondary, tertiary, quaternary. Sickle cell, we've already discussed this. Sickle cell is 1 amino acid mutation of glutamine, got replaced with a valine. This is from your book. This just shows a normal sickle cell. See the glutamine in position 6 got replaced with a valine. The normal shape gives you the nice biconcave disc shape of hemoglobin. The sickle cell shape, the interactions are incorrect, and you get the sickle cell shape. Why can't I say that? Sickle shaped cell. It's a tongue twister. Okay. Denaturing a protein. So proteins have shape. It's important. If I mess with the shape, the protein falls apart or it folds incorrectly. See the picture here? Things that can denature a protein, if I change the pH, changing the pH, we discussed this back in video 1, can change the version of a molecule, like whether it's acidic or basic. So changing the pH can change with the R groups, the protein folds incorrectly. Changing the salt concentration can mess with ionic attractions. Changing the temperature, if I heat a protein, heating things causes hydrogen bonds to break. So if I heat the protein, the hydrogen bonds break, the protein falls apart. Denaturing a protein, to denature means to change the shape. If you, like say you heated it and cause the protein to denature, if you cool it back down, it can renature and go back to its normal conformation unless you went too far. When you cook an egg, you know how the white of an egg looks before you cook it, and after you cook it, it turns white. It turns white because you denature the proteins because you cooked it. you can't renature that back into what it looked like inside the egg. But if I have a protein that likes to be, I don't know, 23 degrees Celsius, and I make it 26 degrees Celsius and it denatures, and you cool it back down, it'll probably renature. So unless you heat the snot out of it, or pH the heck out of it, or salt the heck out of it, you can likely renature it back. Okay, boiling it's probably going to just kill it beyond. But you can't kill proteins. Proteins aren't living. It'll denature it beyond repair. One quick note, X-ray crystallography is a technique that we use to determine a molecule shape. We're going to come back to this technique when we do DNA, because this is a super important technique, especially from a historical perspective. It's how we figured out the structure of DNA. Basically, the idea is you shoot X-rays so you take the protein and you crystallize it, which is kind of hard to do sometimes. And you shoot X-rays at it, and you see how the X-rays diffract or interact with the protein, and you get a pattern on the X-ray film, and you use the patterns to figure out what the shape of the protein was. You can do this with proteins. You can do it with DNA. It's not just for proteins. But yeah, just kind of file X-ray crystallography for later. Okay, we've done 3. One more. So there are chapters in your book on DNA and nucleic acids. So we're just going to super skim the surface here, hit the highlights, because we're going to revisit this later. So there's 2 kinds of nucleic acids, DNA and RNA. DNA is deoxyribonucleic acid, RNA is ribonucleic acid. What the difference is, we're going to see in a minute. They're called nucleic acids because they were first discovered in the nucleus of a cell. DNA is always in the nucleus. Well, there's always an exception, but DNA, your chromosomes, your genes don't leave the nucleus. They leave the nucleus, something's the matter. RNA does leave the nucleus. So finding RNA in the cytoplasm is normal. Finding RNA in the nucleus is normal. Finding DNA in the nucleus, normal. Finding DNA in the cytoplasm, again, that can happen. DNA can be elsewhere, but that's not the general theme. So the parts of a nucleic acid, the monomer is called a nucleotide. And the 3 parts of a nucleotide are a nitrogen base, a phosphate group, and a 5-carbon sugar. So this is a nucleotide. No, I don't. So here I have a phosphate group. Here I have a sugar. In this case, it's pentose, a 5-carbon sugar. And here I have a nitrogen base. Nitrogen bases are called that because they contain the element nitrogen, which is, I know, a shock. The word nucleoside, this isn't that big a deal, but if you have a nucleotide without the phosphate group, you call it a nucleoside. Note one thing about this picture. See how the 5 carbons are like, that's the little apostrophe is prime. So that's 1 prime, 2 prime, 3 prime, 4 prime, 5 prime. We label the carbons with this notation with a number and the prime sign. So like that's the 5 prime carbon. The 5 prime carbons attached to the phosphate group. The 1 prime carbons attached to the nitrogen base. DNA versus RNA, the 5-carbon sugar in DNA is called deoxyribose. In RNA, it's called ribose. The only difference is, see this carbon number 2 in DNA, it's just an H. In RNA, it's a hydroxide group. So, deoxy just means you're missing an oxygen, deoxy. So deoxyribose is missing that oxygen on the 2 prime carbon. So there really isn't much of a difference between DNA and RNA in terms of the sugar. It's just 1 oxygen group. The nitrogen bases, you've done this before. So there's 5 of them total. There's only 4 in DNA and 4 in RNA. Adenine, guanine, cytosine, thymine, and then uracil in RNA. Purines are big boys. Purines are adenine and guanine. They're 2 rings fused together. Pyrimidines only have 1 ring, cytosine, thymine, and uracil. In DNA, you have A, G, C, and T. RNA, you have A, G, C, and U. In RNA, there's no thymine. It's uracil. And again, this is a later chapter. We'll come back to this. If you look at the difference, that's uracil, that's thymine. Where is the difference even? That methyl group right there is missing in uracil. So the difference is only a methyl group. It'd be nice if they would just use the same thing. So we didn't have to deal with the letter U, but in RNA, we had to deal with the letter U. Okay, cool. So you've seen the structure of DNA before. So it's a single strand piece of DNA or RNA looks like this, where I have a sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate. And each sugar is connected to one nitrogen base. All right, it could be a purine or a pyrimidine. Okay. Notice my carbon numbering system. 1' carbon goes to the base. The 5' carbon goes to the phosphate group. And the 3' carbon is not shown here, but this 3' carbon, if you go back, is attached to a hydroxide group. Okay. This is the same thing, just bigger. So sugar, phosphate backbone, and the rungs are the nitrogen bases. We did that. So I'm gonna skip this and come back to it. So if you look at which one do I want to show? If you look at, actually, you know what? We'll just do it here. So we discussed the prime notation of carbons. So when you join nucleotides together, so just look at the picture. This 5' carbon is attached to a phosphate group. All right, that phosphate group is attached to the 3' carbon of the next nucleotide. All right, now keep going. That nucleotide has a 5' carbon. Look where I'm pointing. Goes to a phosphate group. The phosphate group goes to the 3' carbon of the next nucleotide. So the sugar phosphate backbone involves the 3' carbon, which is this one, and the 5' carbon, which is this one. So if this nucleotide was going to form a piece of DNA, you would find a bond between this 3' carbon and a phosphate group down here, okay, which is what's showing in this, this is four nucleotides. The whole 5' to 3' deal, which is how DNA polymerase copies DNA, you probably remember from freshman year or whenever you took biology, how polymerase goes 5' to 3', that involves these same numbers. For right now, just understand that DNA has a direction. In this case, here's the 3' end of the whole thing, here's the 5' end, okay? This is super fuzzy. This just shows the same thing. RNA is usually a single strand, so like this could be a piece of RNA or a piece of DNA. DNA is usually a double strand, so you have 2 pieces of DNA that are wrapped around in a double helix, and the 2 sides are anti-parallel, which means if one goes 5' to 3', the other one goes 3' to 5'. And you can kind of see in this picture, this first strand begins with the 5' end, and this strand begins with the 3' end. Again, we're going to come back to this when we do DNA, but the term for that is anti-parallel because they go in opposite directions. Base pairing rules. So because DNA is a double helix, the nitrogen bases, the rungs bond together. A always pairs with T, G always pairs with C. Here I have a C and a G, here I have an A and a T. The bonds that hold the DNA double helix together are just hydrogen bonds again. So, you take 2 strands, form the double helix. That's held together by hydrogen bonds, which remember, hydrogen bonds are very weak bonds. They're not that strong. But if you have a lot of them, they have a big impact. So imagine a chromosome or a helix that's, I don't know, 10 million bases tall. So that's 10 million bases bound together with hydrogen bonds. Between an A and a T or G or C is either 3 hydrogen bonds or 2. I think G and C has 3, A and T has 2 between them. So that's many, many, many millions of hydrogen bonds holding the helix together. If I wanted to break the double helix, how do you break hydrogen bonds? You could just heat it. All right, heating hydrogen bonds breaks them. So if I heat DNA, it's going to fall apart. Not the backbones won't fall apart, but the strands will stop base pairing together. In RNA, there is no thymine. So in RNA, A pairs with U, not with T. So if you're transcribing a strand of RNA, you should never see the letter U. I'm sorry, you should never see the letter T. Where there's a T in DNA would be a U in RNA. Okay, this just shows all that a little bit bigger. So to wrap it up, your textbook has these 2 diagrams which go through carbs, lipids, proteins, nucleic acids. This is a great review if you want to see them, everything together on one page. This is from your book. Okay, that was a lot in one video. That's a super place to stop. So hope that was helpful, and I will see you guys next time.